Okay, good afternoon, everyone. We will continue where we left. In the last class, we had a good introduction about splicing, where it occurs, and the basic principles or the basic concept of it. And today we will try to go into a little more details. And I'm not going to take up the whole lesson because it will be too much and we cannot go into so much of depth. But we will still cover a few things so that you will have a better understanding of what you are learning. So eukaryotic genes, most of them are interrupted to mean that they have introns between coding regions. This would be the start codon, and the last one could have been the stop codon. And within those, there are non-coding regions within the gene, and they are called as introns. Eukaryotes, if you see with increasing complexity, you will find more and more number of introns. So whether it just happens or whether it is required, whether it has a purpose, whether introns have a purpose is still under debate. So typically, transcription would occur, and the pre-mRNA is produced, and then the exons are spliced together to generate a mature mRNA with all the exons, and the introns are excluded and eliminated, finally degraded. Transcription and splicing occur within the nucleus. And some more things to remember for eukaryotic mRNA is about the G cap on the 5' side, methylated guanosine, and you have a 3' side poly A tail on the 3' side of the mRNA. So this is where we are talking about. There are approximately on an average, there are 7 introns per gene. If you look at complex organisms, including plants or animals, it doesn't matter. We also learned about the borders between the exons and introns, like in this case. I don't need you to mug up these, but it's all right. It's good to know that there is a conserved sequence on the 5' splice site and also the 3' splice site. As we have seen in the previous one, there is also a branch site. There is a conserved sequence within which this adenine is important for splicing to occur. And we saw the trans-esterification reaction where an ester bond is formed and another one is formed. One is broken and another one is made. So we call it as trans-esterification reaction. So we also discussed about what is a lariat. It is like forming a rope-like thing like this that is shown here, and there is only one end that we can see, that is the 3' end. The 5' end is covalently linked with this by forming a 3-way junction, right? We also discussed about a 3-way junction. This is you would be having, this is the RNA, and here is where the junction has happened. This is 3' OH of adenine, and we have here is the 5' OH, right? Sorry, this is not 3', this is 2'. Okay, that's where we are wrong. 2' to 5' linkage in this location. And we also discussed about spliceomachinery. There are many. This is especially true in the case of eukaryotes and nucleus. Again, within eukaryotes, there are 2 sets, right? 3 different. If you take a plant cell, for example, there is DNA within the nucleus. There is DNA in the mitochondria, and there is also DNA in the chloroplast, right? So what happens? Splicing that happens within the nucleus is different. And within mitochondria and chloroplast, it is a little bit different. So what I'm discussing here, the spliceosome machinery, is mostly about the nucleus, nuclear DNA, right? It has about so many proteins, 150 proteins, and so many RNAs, like say 5 RNAs. It's an active process, and the RNA components are involved in major functions of catalysis as well as recognition of the borders, 5' splice site and 3' splice site. This is how those recognition happens. Say here is a ribonucleoprotein, small nuclear ribonucleoprotein, or SNRPs, you can say. They have the RNA sequence. They have to recognize a sequence here. And to do that, they involve a component that is the RNA component, and the protein component facilitates this interaction between the 5' splice site. And the same sequence is recognized by later at a different protein. Say, for example, at one position, at one point, it is recognized by U1, and a later point, the same sequence is recognized by U6. And how is that useful or better? It's because, say, for example, if UN might make a mistake. Say, for example, if there is something to be read, okay, something is here, and if one person is reading it aloud, and there could be some mistakes, right? And here, suppose you are here, and who is listening what is being said? You cannot trust it as much. But first, this person has read, person A, and left, and then you have another person to come, and the person is reading the same thing. And if these two match, then you would believe the information to be true, that the information present on this paper is correct. It's almost the same way. What you're seeing, you can think of the paper here as the 5' splice site. It has to be, if it is read by two people, then it is likely more accurate. If it is just being read by only one person, there could be mistakes, right? That is how we will see the pathway. But at one point, U1 is recognizing, at another point, U6 is recognizing the same 5' splice site. So the fidelity increases. It's unlikely that both will make mistakes. Okay, there is still a probability, but the probability would reduce. I hope you understood what this means also. Okay, so many sequences are recognized by multiple proteins like this. So we have already seen these things, what happened, nuclear pre-mRNA. There are 3 points, 3 ways of how things happen, or the classes. Nuclear pre-mRNA, that happens mostly within the nucleus, most eukaryotic genes. And the chemistry is almost the same. There will be subtle differences, and we will discuss them. We already discussed, we will discuss them again. So, group 2 and the spliceosome machinery, or the nuclear introns, they undergo by similar mechanism. Yes, sorry. Group 2, and the chemistry is same. There is a branch point A adenine, and using its 2' OH, there will be a nucleophilic attack, and the 3' side of the 3' OH of the exon makes a nucleophilic attack, and you will have the splicing of these 2 exons, and a lariat is released out. The same is the case with nuclear introns, except that there is a complex of ribonucleoproteins that are involved in mediating this. In group 2 self-splicing, the phrase itself, self-splicing, they do not require these accessories. Instead, they undergo it by themselves. Group 1 splicing, self-splicing, is a little bit different, except that it coordinates all kinds of makes a microenvironment where a guan in GTP is present in there, and it is properly oriented such a way that its 3' OH can be activated, and that 3' OH will make an attack on the at the 5' splice site, and this is the G that now is covalently attached. And you have the 3' OH of the exon, which will now be bound spliced with the subsequent exon, and that is how splicing occurs. In terms of exon splicing, it's almost the same. You need to have exons in continuous readable frame by the ribosome. The products will differ. In nuclear spliceosome and group 2 self-splicing, you have a lariat formation, whereas in group 1 splicing machinery or self-splicing introns, you find a G kind of cap-like thing, right? So this is 3' OH, and you have a G on the 5' side. Let's look at what happens in the nuclear ones and in coordination with nuclear proteins. Spliceosome mediated splicing. Actually, the whole of these things you can call off as a common name, like spliceosomes, like a name of ribosome, for example. So this is the 5' splice site, and this is the 3' splice site. Okay, and we have a branch point. We already know that. So U1, as we saw in the previously, it recognizes the 5' splice site. BBP recognizes the branch binding protein. You can say branch point binding protein. That recognizes the sequence here, and it binds there. And on the 3' splice site, you have U2AF65 and 35 complex. Okay, and then that is recruited by say BBP is released, and U2 SNRNP binds there, right? So you can think of the branch point is now proofread or recognized by at least 2 proteins. First by BBP, and second by U2. Similarly, you can also think of U1 at the 5' splice site. It is first recognized by U1, and now you have a tri-SNR RNP particle consisting of U4, U5, and U6 that binds to the 5' splice site and also the branch point site. That is, by recognizing or by interacting with U2A1, they bring these 2 points close together. Okay, and now U1 is released out, and the 5' splice site is now recognized by U6. U6 can also, in a different part, can interact with the branch point as well at some places. So the first cut will be made. I mean, the 2' OH of adenine will make a nucleophilic attack, and then there is a trans-esterification reaction from here to here. And the exon 1 and exon 2 are now continuous or spliced together, whereas the lariat intron is removed along with the spliceosome components. Okay, so it is simple to remember, and again, if it is an exam, it's a good idea to always have a look at it. So to start with, U1 on 5' splice site, BBP on the branch site, and U2AF6535 at 3' splice site. BBP is replaced with U2. Then you bring in the tri-SNP RNA nucleotide, SNRNP particle, and then U2AF6535 is removed, and these 2 are brought in together, and then you have U4 released, and this trans-esterification reaction happens, and then the lariat is released out, and the exons are spliced. That's what happens in spliceosome mediated splicing. The similar interactions also happen with group 2 introns. I'm not going into the details of it, but what is important to realize is that in group 2 you have U2 in the spliceosome mediated ones you have U2 which has an RNA component. There is also U6 which has an RNA component, right? Whereas U2 is here binding to the branch point after BBP. And then we also have U6 that interacts also with this region. So what I'm intending to say is these are separate components in spliceosome mediated splicing, whereas in group 2 introns they are self-splicing. They do not require any additional components. Everything that is required is within itself. So within the intron, there are domain 6 and domain 5, which fold a little bit similar to that of the U2, U6 regions. And then they interact, and the branch point will make an attack and will bind here. So there'll be a nucleophilic attack, and the cut is made here, which will be a transesterification will happen between these two then. Right? So the important thing to understand is RNA folds upon itself to create microenvironments such that there can be some bases can be activated, and then some reactions like transesterification in this case can happen. It's almost like an enzyme. That is the ribozyme. If you talk about the ribozyme, one second. Something is freezing, I guess. Yes. Sorry. I think it's not playing. Okay. So in group 1 introns, it's almost the same, except what we need to remember is the G here. This G is not part of the sequence, but it is by folding, by creating a microenvironment, the RNA will put a GTP, whose 3' OH could be activated, and that 3' OH activated will make a nucleophilic attack and mediate the splicing of the exons. So we will discuss about it some other time, because there is a video which I would like you to watch it before we can discuss. But otherwise, this is not a big difference. Okay, now it's forming. Yeah. So transesterification reaction is you break a phosphodiester bond and make another one, right? An acid group and OH. So you have, this is the 3' OH that I have indicated here, and the 3' OH is activated, and that makes a bond here. Then now you have the G is added to this part like this, forming something like this, and the exons, the exon, the 3' OH here will then make an attack on the next exon to form spliced mRNA, spliced exons basically. So what is the fidelity of splicing? What do you mean by that? The accuracy with which it happens. So there are two, or at least two kinds of mistakes that can happen. One of it is called as the exon skipping. So if this is the 5' splice site, okay, and it has to splice or bind with another 3' splice site, but there is also another intron here. So you have another 5' splice site and 3' splice site. To mean that The first 5' splice site can bind here like this, or it can also bind to another 3' splice site down the line. So if this is what happens, this exon is skipped, right? And that is, so if you have, this is a incorrect mRNA consisting of exon 1 and exon 3, like is shown here. In the B, there is another kind of mistake that can happen is if there is a sequence. We discussed that there is 5' splice site and there is 3' splice site. It has a conserved sequence like that. Sometimes some sequences within it could be within the intron, or it could be within the exon, that might resemble that of a 3' splice site. And in that case, what may happen is the exon here might splice with this one, which should actually happen here. This is the correct one, and this is the wrong one, but it may happen. There is always a possibility, and those sites which resemble that of the 3' splice site are called as pseudo splice site, right? So there are two types of errors: exon skipping and pseudo splice site selection. How is fidelity ensured? As we learned in the last lesson about transcription, when we were learning about it, we learned about the promoter escape, we learned about elongation, and so on. And while learning that, we also learned the CTD, carboxy terminal domain of this RNA polymerase 2, which is long extended. It has several sequences of these, something like this, and they can be modified by phosphorylation. And the modification tells something. So basically, if it is if something, this serine is phosphorylated, promoter escape will happen. Once the promoter escape happens, capping protein will bind. If we take the mRNA, while discussing that lesson in that lesson too, I have discussed what it means by this one. So we have an exon, intron, and then we had several poly A tails. Say, for example, 1, 2, and 3, and these are the introns. If RNA polymerase is moving like this, what is the first thing that would come out? That will be the 5', and the first thing that we need is capping. So the phosphorylation here will increase the affinity for capping protein. So the capping enzyme will join here, and as soon as the mRNA comes, this capping enzyme will jump onto that. And what is the next thing that would come? We may have start codon that has nothing to do. Once this part has come, that would be the 5' splice site, right? During that time, we have once the capping enzyme is released, this phosphate group is removed, and a serine at second position is phosphorylated, and that will recruit the splicing components. And first, what is coming out is the 5' splice site. So there you will have U1 and other proteins coming and binding. Then we have the branch point that is coming. So you have the BBP, branch point binding protein, coming and binding. And then we have the next one that is going to come out is the 3' splice site. So the order in which they are coming, we will have these proteins coming, these machinery, splicing machinery binding appropriately, and then splicing already trying to occur. Okay? So that is what usually happens. But that's not all. There is also another way of how splicing machinery, splicing fidelity is achieved. That is by there are several sequences that is called as, first one I would like you to focus is the exonic splicing enhancers. These are sequences within the exons, and they are present within the exons, and they have affinity for proteins called as serine arginine rich proteins. Those are called as SR proteins. So basically, if you see from here to here, that's the exon. And the exon has multiple ESCs, and which would mean that the whole exon is coated, kind of coated with SR proteins, right? That is what is meant by exon definition, in the sense that the coating of SR proteins here will say that this is where the exons are there. And then the next thing, what it does is SR proteins, if they are here and nothing else is here, then they will recruit the 3' splice site machinery. On this side, they would recruit 5' splice site machinery, right? But of course, I hope you recognize that this is the intron from here to here. And then we call this as the 5' splice site. And this is the 3' splice site. We have a branch point here. Okay. The meaning, what I'm trying to say here is the exons, within the exons, there is a way to prevent any faulty splicing happening within the exons like this. That is because of exonic splicing enhancers, and because these exonic splicing enhancers have affinity for SR proteins. And SR proteins have dual function. One is because this exon is already occupied, there is no more space for any splicing proteins to interact here. Second thing they do is on the 3' side, they will allow the binding of 3' splice site machinery. On this side, I mean here, whether you talk about exon or intron matters, right? On this side, it is about 5' splicing machinery, and this side about the 3', they will recruit 3' splicing machinery. So that's a kind of, it increases accuracy and also efficiency, right? Yes. So there are some variations to the splicing, and one of it is there are two that we will discuss, and that is interesting for, it's called as trans-splicing. So far what we have discussed, excuse me. So far what we have discussed is that splicing always happens within a single RNA molecule like this. And that gets converted into something like this. Spliced exons and the intron is left out. That is one RNA making a mature RNA. But trans-splicing is splicing happening between two different RNA molecules. Here is RNA1 and this is RNA2. And it follows the same thing as group 2 introns basically. Group 2 introns use branch point adenine like this. Group 1 introns use the microenvironment created for the GTP, non-covalently attached. And finally, of course, I hope you remember that. So trans-splicing is almost same as group 2 splicing, self-splicing machinery, except that it is happening between two different RNA molecules. So the 5' splice site here is will go and make will bind with the branch point, will form a 2' 5' linkage with the branch point of another RNA molecule, a different RNA molecule. So normally here we would have gotten a lariat like this, but in this case we won't have a lariat. Instead, something like a Y is present because it is forming between two RNA molecules. The end result is same, but we will come to a discussion where I think now and then I keep reminding you, one gene produces one protein, one gene. We'll come to that slide too. But with trans-splicing, you can think of it like this. Two genes giving rise to one protein. That's amazing, isn't it? That's another level of complexity. They're not very often, they're not very often present. I mean, they're rare in a way, but it's interesting to know that there is a possibility of it. There is another variant to splicing which is called as ATAC, spliceosome machinery, which is called minor. The what we have learned so far, spliceosome machinery is the major one. Yeah, initially they were thinking it is 5' AU at the 5' site and AC at the 3' splice site. But eventually, most of them have turned out to be something like GU and AG termini at the 5' splice site and the 3' splice site. So they have almost the process is almost same, except that there are differences in the components a little bit of these. You have a different variation of U4, U6, and so on. But otherwise, the machinery is same. So I'm not going to discuss that much. Also, they must have. So you have group 2 introns. We learned about spliceosome machinery. Then this is another one which we say is that ATAC spliceosome machinery. The chemistry of transesterification remains the same in all the three. So it's likely that these two have arisen from group 2, because this is what we see in prokaryotes or primitive ones. So eukaryotes have emerged or evolved from prokaryotes, so it's likely that these came later. But it is still a question somebody can debate on. This is what I was trying to discuss. Initially, people talk about, when we say one gene, one character. Something like our skin color, for example, is produced by melanin. So one gene, one character. People used to think it that way. Later, they decided, they evolved. No gene by itself can give rise to a character. It's a philosophical way of saying it. Without the ability to say, for example, I have a gene X, and it gives, say, color, some color. This gene first should be expressed, right? You need the transcription machinery, translation machinery, and only then you will have a protein. And this protein will, or it's an enzyme, say, for example, and it converts something to something. And say substrate A to product B. If this is what the enzyme X does, where is A coming from? Right? So that means what I'm trying to imply is, it's not, never there is a situation where a gene that is present just by itself can give rise to a character. It's always, it has to be in context. And what we now agree is many genes can give rise to one character. When you say one character itself is a multiple things actually. And it requires, one character can require many things to happen. Say, for example, skin color, then means you have to acquire with ability to produce melanin, and then only in the melanocytes, and so many things like that. So people have thought about, we need to bring a statement that is kind of global and true, and like the central dogma we say. People try to bring about this. Then they came up with one gene, one enzyme. So one gene can only produce one enzyme. And then we now know that enzymes, like, say, for example, RNA polymerase, it's one enzyme complex. It consists of multiple subunits. So it has a quaternary structure. So then people reduced, here it is that not all the genes produce enzymes. There are structural proteins, something like actin, for example, or microtubules. If you see, they are proteins. You can talk about any membrane channels or nuclear pore complex proteins, if there are any. They are structural proteins. They are not necessarily enzymes. So then they came up to this, one gene, one protein. And then you have one protein can be made up of multiple subunits. So they came to one gene can code for one polypeptide. one polypeptide, the difference is when you say polypeptide, it is a continuous stretch of amino acids, right? And as we saw in the previous slide, like in splicing, it's like two genes can give rise to one polypeptide, because one gene give rise to one mRNA, another gene give rise to another, which has exon 1 and exon 2. And because of splicing, you produce a new mature mRNA with exon 1 and exon 2. There are two genes giving rise to one protein or one polypeptide. And not necessarily that all the genes produce proteins. Some produce only RNA. So this is the kind of thinking one must try to see. Why I'm discussing here is what is the probability that one gene gives rise to many proteins. One gene gives rise to many proteins. If you take something like the coronavirus, it produces one large polypeptide, and that polypeptide, okay, it is producing one huge protein. Say I'm trying to draw it as a protein, and then there will be a cleavage within themselves like this, and it will produce, they will become smaller proteins, and each has a function. This is present in many viruses and so on. So here, one gene is giving rise to many proteins. And there is another way of how one gene can give rise to many proteins, and that is by alternative splicing. The essence of alternative splicing is something like this. So here, if you take one gene which has exon 1, 2, and 3, and it has two introns, intron 1 and 2. Then upon transcription, we have the production of a primary RNA transcript. And then there are at least 5 ways of how things can happen. First thing is what we consider normal, where exon 1, 2, and 3 are spliced together to produce a complete mRNA with a complete coding frame, reading frame, start codon to stop codon. In the second scenario, what happens is, I hope you remember we already discussed this exon skipped. There we can see it as an error. Here we can say it is deliberately skipping one of the exons. Here exon 2 is skipped. Splicing between 1 and 3 exons will result in a smaller protein, but it could have its own functions, new functions. In third one, there could be a pseudo splice site within the intron, and then you have intron 1, 2, and 3, but the small part of the intron is also left. That is referred to as exon extended. In some other cases, you have the whole intron itself retained. Sometimes splicing may not have occurred, and the whole intron is retained. And in what you can say is alternative exons, say here exon 1 and 2 could splice and give rise to this, and exon 1 and 3 could be spliced to give rise to a transcript like this. This is what I'm now going to say is not absolute, but I think it will give you something to think about. When a ribosome is reading, okay, within the open reading frame, what it needs is AUG, that is the start codon, and a stop codon, TGA or TAA or TAG. And this reading frame should be continuous. Codons should be continuous. There are variations to what I'm saying now with regard to coronavirus. When it comes, we will discuss, and there are many other examples. But otherwise, normally a ribosome would bind and start translating it. So the first exon should have the ATG. If splicing occurred between 2 and 3, and you have a transcript like this, 2 and 3, and if there is no start codon here, translation would never initiate, and that becomes a non-functional one. Similarly, if splicing happened between exon 1 and exon 2, and there is no stop codon here, then ribosome might end up translating further until it encounters a stop codon. At that time, also, you might have a dysfunctional protein. May have it. Okay? So I hope you understand that in all the cases, exon 1 is included. Why is that? It's because you need a start codon. And in all the cases, you see that exon 3 is included too. And just making up, this is a theoretical one, but it's good for thinking. In this case, exon 1 and exon 2, this transcript could work well if there is a stop codon to this in the end of the exon 2. So we'll see a few examples and a couple of examples, and then we will stop. Alternative splicing by troponin T gene. Troponin is a protein that is involved in muscle contraction. There are 2 different enzymes that are produced. Before I go further, I think I have to also describe this. So while I just give me a minute, so I will just. Yes, here are 2 words that I want you to remember: constitutive and regulated. Constitutive means it is continuously happening. Regulated means we will decide, or the factors that are present will decide whether the splicing should happen or not. I will take the example of the 5th case here. Constitutive means this transcript is produced continuously, and some of the transcripts will fuse, will splice like that, 1 and 2, and some transcripts will splice 1 and 3 like that. That is constitutive. No factors are involved. It is a randomly whichever happens. When you say regulated, if there are some other factors involved, if this factor is present, only 1 and 2 will be present. 1 and 2 transcript will be produced. If this factor is not present, then only 1 and 3 will be present. That means there is regulation of the splicing, which one should happen or not. So one gene is giving rise to, say, for example, based on some of these, at least 2 or 3 different types of proteins. They are typically called as isoforms. Iso is same, form is something like structure. It's not necessarily true, but what you are seeing is there is some, in these 3, if you count, there is certain degree of similarity. They all have exon 1. That means that protein, when it is transcribed, that protein sequence will be common among these. They all also have the exon 3, right? So there is certain degree of amino acid sequence similarity. Similarity means actually it's a kind of identical. 3 and 1, the sequences are identical. What is varying is within the whether 2 is present or not, and whether there is any addition or not. Okay, so they may have, those are called as isoforms. They may have similar functions. Sometimes they may have distinct functions. Sometimes they may have antagonistic functions, but also they may have non-functional too. Here is the example of troponin T gene, and this is the primary transcript that is produced. It has exon 1, 2, 3, 4, and 5. It's made up of 5 exons. And through alternate splicing, alpha troponin could be produced. If 1, 2, 3, and 4 is excluded, then the protein that forms is called as alpha troponin T. It has a function as well. And there is another way of splicing, that is 1, 2, 4, and 5. 3 is skipped now. Either of them will happen, both will not happen, right? You will have 1, 2, 3, 5, or 1, 2, 4, 5. And based on that, you're generating 2 different two distinct proteins with related functions because they share a high degree of, say, domains. At least in terms of domains, they share similarity in this, or identical in amino acid sequence. And the constitutive alternate splicing, another example is of SV40 virus. So it has exon 1 and exon 2. And what happens here, there is 5' splice site. There is a stop codon here. It is relevant here. Stop codons can be present anywhere, okay? Anywhere within the intron, because first of all, we don't know the reading frame, right? And there is also a 5' SST and a 3' SST. So there are two ways of how splicing can occur within this SV40, this T antigen. One of it is that splicing occurs between this 5', this is something like a pseudo splice site. Yes, 5' splice site here, and they will, this is how splicing would occur. So the majority of the intron is actually within the reading frame. And it produces a smaller protein because ribosome starts here and it will start translating. And it keeps on translating, and it now encounters a stop codon. The stop codon is within the reading frame of this protein. And that's why we mention it as stop codon, because it has relevance here. Because of which also it is, the ribosome is never going to read the rest of it, because it's going to, termination will happen here, translational termination. When we read translation, we will get into it. So it produces a small T antigen. There is another way of how things happen. That is, this is the 5' splice site. It is making a legitimate splicing with the 3' splice site here. And now you have a production splice, a mature mRNA of consisting of exon 1 and exon 2. So the ribosome would bind here, and it will keep on, it will read exon 2, and it will also read the exon 1 and then exon 2, producing a larger protein. So this is called as capital T antigen, and the other one, the smaller one, is called the small t antigen. They both are functional. They have similarity in the based on the exon 1, and they have function. The capital T, it induces transformation. I hope this is the second time, I hope you remember that this is the second time we are using the word transformation when it comes in molecular genetics course. The first one we first learned about it is when we were discussing about Griffith's experiment. There we said about transforming principle, where a rough strain becomes smooth strain, right? And this transformation here, which would mean that a normal cell becomes cancerous. Okay? And here it means the normal cell becomes cancerous. And the one we learned in the beginning of this course is about how it's about uptake of naked DNA and changing the characteristics. Okay, just telling you so that you don't confuse. So T cell induces transformation and cell cycle re-entry. That means there is the cell that is infected with SV40 would lose its ability to regulate its cell cycle. That means, what is a cancerous cell? It continuously divides, forming a tumor, and that is cancer basically. The small t also has a function, T antigen. The cell has two things. One is to grow and divide, okay, divide, and another one is to die. Death. Capital T increases the ability to divide, and small t reduces the ability to die. That means the cells will continuously divide and never die. So those two together are actually going to cause a problem, to cause cancer basically. And whether this one is produced, capital T or small t, is also dependent on some SF2 and ASF concentrations and so on. Okay, so I will stop here because there are a few things that we will discuss in the later class. And today is the deadline for the assignment. I hope you are, I will just stop the recording.